when I'm in the classroom with the second graders, I am way more intimidated with what's going on in the classroom with the second graders than with any of those other kinds of classrooms. And probably for many of you, a second grade classroom would also be intimidating. But I have some appreciation of the level of energy that it requires to really teach in the schools. My work focuses in the laboratory over at the medical school on the immune response to bacterial pathogens, and in particular what kinds of bacterial host cell interactions result in stimulation of various arms of the immune response. And I will talk a little bit about innate immune responses, those responses that are not specific to the kind of infection that you're getting, and then to transition to what's known as the adaptive immune system, the part of the immune system that has a memory and where the re-encounter with the pathogen causes a swifter and more robust immune response. And what I'd like to do is use the chalkboard for the entirety of the lecture, and I really, really hope that what you'll do is that you'll interact and yell out questions when you have questions as we go through the immune system, as well as some of the pathogens that we'll talk about today. And we'll see how it goes in terms of time. But what I'd like to do is first talk about general principles of the immune system and how the immune system responds to infection, and then go and talk about the different arms of the adaptive immune response that responds, and then from the adaptive immune system, talk about how the adaptive immune system really is involved in resolving various kinds of infections. And I'll talk about Listeria monocytogenes, a foodborne pathogen, as an example, as a model pathogen. But I'll bring in some other pathogens as we go through as well. And again, the research in my lab focuses on Listeria, this foodborne pathogen, on chlamydia, which is a sexually transmitted disease in the developed world, and in the developing world causes an eye infection known as blinding trachoma. And it's Salmonella, another foodborne pathogen. And if we have a little time at the end, I'll talk about some of the work that we're doing that's related to stimulating immune responses, where it has applications in terms of vaccines and what kinds of concepts one needs to think about when designing new vaccines against some of the pathogens where we don't have vaccines. So what happens when a host, whether that's a mammalian host, a mouse, or a human, gets infected with a pathogen? There's really two outcomes that are possible during an acute infection. Either the pathogen wins, its growth rate exceeds the capacity of the immune system to contain it, and the host, the human or the mouse or the animal, dies of infection, or the immune system is able to suppress that infection to the point that the pathogen is driven out of the host and the host lives and the pathogen dies. All that a virus wants to do is become more viruses. All a bacterium wants to do is become bacteria. It's the measure of evolutionary fitness. These microorganisms just are wanting to replicate and spread to a new host. That's the measure of evolutionary fitness for these kinds of pathogens. So when the pathogen enters a host, one can look at the pathogen level as a measure of duration of time during the infection. And the first part is entry. And we talk about that in the time frame of around 0 to 4 hours. And so what protects the host during those initial encounters with the pathogen are in large part barrier functions, things like our skin that prevent the easy entry of organisms into our bloodstream, things like coughing and the respiratory mucus that drives organisms up and out of our respiratory system, and other barrier functions in our gut and our genital tract that are constantly flushing pathogens out of these areas where they might enter into the host. And so those barrier functions are one important level, and then just underlying those mucosal surfaces where the pathogens often enter are phagocytes, specially designed cells that take up, gobble up, and destroy the microbes as they first enter. And so this early or entry phase is really in the first hours, and those often clear the pathogens, and there's no even lasting impact on the host's immune system in terms of that encounter. A little later, in the So both are underlying these mucosal surfaces, as well as special cells known as dendritic cells, or DCs, that we'll talk about, which go on to stimulate other parts of the immune system. So yes, macrophages, dendritic cells, and neutrophils. And so in the period of time just after this entry, there's also elements of innate immunity. And these are all called innate because they pre-exist regardless of which pathogen it is that the host is encountering. But after this 4 hours, some of these phagocytes begin to secrete messenger molecules known as cytokines, which cause inflammation. And so in this period of time here, there's an inflammatory response. And that, again, calls more phagocytes into these local sites of infection that go on to help clear the infection. Yes?
That's the 4 hours on.
So basically, these innate responses are ongoing kind of in the 4 to 96 hour time frame. But once the infection, if the pathogen is successful enough that the pathogen can continue to grow beyond this point, it reaches a point, a threshold of activation of the adaptive immune system where other kinds of cells begin to respond. And these cells are known as the lymphocytes. And so in this innate period, and I'll use this yellow chalk here, and I won't use it very much, but if we didn't have the phagocytes, if these phagocytes weren't present, and we can do this in experimental animals that don't have phagocytes, the infection would go up uncontrolled, and the animal would die. And so if we look at what goes on to clear this infection, what goes on to actually promote the resolution of infection, some of that's the inflammatory response, and some of that is the cells of the adaptive immune system, the lymphocytes. And we'll talk about the different classes of lymphocytes and what their roles are, but what we're talking about here are called B cells and T cells. And these, yes? So if they have lymphocytes and no phagocytes, they will die. That's right. The infection will become uncontrolled. If they have phagocytes and no lymphocytes, what one sees is that the infection comes up and begins to be controlled, but never resolves. And so what's going on here is pathogen clearance, and eventually down to the level of complete clearance of the pathogen. But what's left here, when all is said and done, is what's known as a memory response. And so the B and the T cells that responded to this infection expand in response to that infection. So if you look at the levels of T and B cells in the bloodstream that are responding specifically to this pathogen, it will ramp up, and then it will go down as the organism clears. And we'll talk about exactly how the B and T cells expand. But what's left is this memory population, a low-level but more active, a low-level but bigger population than here that is a larger number of these B and T cells that are able to more quickly respond upon reinfection with the pathogen. And the B and T cells are known as the adaptive immune system or the specific immune system because they have specific receptors on their cell surface. In the case of B cells, these are known as the immunoglobulin receptor, or IG. So if it's a B cell, it will have an immunoglobulin receptor, which is functionally an antibody molecule. And we'll talk about what antibodies do. But these receptors are unique for each B cell or T cell. And the T cell here has what's known as a T cell receptor, which functionally is the same kind of antigen recognizing molecule. It's the part that recognizes the pathogen. And what we start out with and what we develop early in our fetal development is a very, very large set of individual B and T cells, each of which have a unique receptor specificity. And it's one of the wonders of how the immune system works, that through very specific genetic processes and mutation and splicing within these cells as they develop, each one of these receptors kind of has a random but unique specificity. And there's billions of different T cell and B cell receptors that are possible. Yes?
What if you were exposed to something from space?
So this is the great thing about how this works, is that what it's doing through this splicing is generating random receptors that can recognize many, many, billions and billions of different shapes. It's not really recognizing anything based on what humans have recognized in the past. It's just an assortment of random shapes of these receptors that in theory could bind anything. And there's enough of them, given that there are billions of them, that we do a pretty good job of responding to anything that we encounter, even though we have never, and our ancestors may have never encountered that particular pathogen. And
So
yes?
does each B and C have one type of receptor, or are there thousands of different ones?
Each one of them is a clone that has many copies of the same receptor on its cell surface. That's a good question.
Or receptors?
Same receptor
Okay.
in general. There are rare exceptions where through extreme manipulations you could get them to have two. But in general, the way it works is that each B or T cell has one unique receptor specificity. And yes?
Sorry, just you were saying about from outer space, there's no kind of, it doesn't matter if you've never seen it before, but is there anything that gets passed on genetically in terms of receptors that work well?
No.
No? Okay, so
There-
it's all random for every individual when they start.
That's correct. Now, what those are, we're going to talk about what the T cell receptor and what specifically the T cell receptor recognizes in terms of how it binds to its antigen. And there's a genetic component to that. But the answer to your direct question is that we don't pass on any receptor knowledge or any knowledge of what you encountered. Your children will not have an immune system that's different because you encountered a particular pathogen unless you continue to be infected when you're sharing a bloodstream with them. So what that means, though, is having all these different shapes, possibilities of binding in any different shape, is as we develop early in development, we also have the danger of recognizing our own cells and our own proteins within our bodies. And so there needs to be what's known as clonal selection to get rid of shapes that respond to self. And so what the immune system is doing is not responding to foreign things, to non-self. What it's doing is not responding to foreign things, it's responding to non-self. I could say that again because I didn't say it so clearly. What the immune system is not doing is responding to things that are foreign. It's just responding to things that are not self. And so how does it get rid of self? When the B and T cells are made, the B cells are made in the bone marrow. The T cells are also made in the bone marrow, but they undergo education or maturation in the thymus, a gland in the neck area, the upper chest. And what happens during education, the B cells are educated in the bone marrow, the T cells are educated in the thymus, is that T cells or B cells that recognize things that are around them in those environments get activated and undergo programmed cell death. So that these cells are engineered to self-destruct if, when they first develop, they're recognizing their immediate surroundings, which is self. So everything that's left is potentially able to recognize non-self.
So a person with an autoimmune disease,
Fails in that process at some little.
why doesn't it happen right away? Why do people not get MS until they're...
And that's one of the great mysteries of autoimmunity. Is I wish I had a good answer. Right. So one of the things that people with MS are responding to are particular proteins in the nerve sheath, and it may be that there's physiological changes in the structures in the body that allow exposure of antigens that aged and are therefore different. It could be that the encounter with a pathogen, what I'm going to tell you is that these expand in response to pathogens. And that it may be that a very low-level response isn't meeting the threshold of activation, but that if you have an insult with a pathogen that might closely mimic the shape of the nerve sheath cells, that those, as they expand in response to the pathogen, may now overcome the threshold that are then able to self-react.
But some of those diseases occur in families.
Some of those diseases occur in families. And we'll talk about, again, the genetic component of how one of the genetic components of how the immune system is controlled. And so it's really a disease of T cells, and T cells, the T cell receptor responds to a molecule on the host cells that does have a familial component, a genetic component to it. Yes?
So the apoptosis occurs in the bone marrow and the thymus before the cells are released, or?
Yes.
But I mean, how do they find out what's certain and what's not? I mean, how do you distinguish, how do you do the clonal selection if they haven't been exposed to anything?
So
If they're in,
all it's doing is depriving the repertoire, the total number of T and B cells, the total number of lymphocytes. All it's doing is reducing the repertoire because of self. It doesn't, the selection is occurring only after the pathogen is encountered. So it's negatively selected in the way the lymphocytes work is if they see self, they're deleted. They also have to recognize, they also have to have some sort of functional capabilities. They're programmed to have some reactivity at a low level, which allows them to be what's known as positively selected. But the biggest thing that goes on in the thymus and the bone marrow is that the things that are around them that represent the self that it might see are those clones that recognize those antigens are deleted.
everybody else, all the other cells that are in the
That's a really, really good and sophisticated question, and what the answer is it's not good enough. And so, because you could imagine that it doesn't see toenail cells in the thymus. And so there's proteins that are specifically in toenail generating cells that aren't in the thymus or the bone marrow. And so there's a second layer of protection called peripheral tolerance, where we don't activate these T and B cells unless there's a second danger signal in that area. So one of the roles of these cells here, the phagocytes, when they encounter pathogens and set out these cytokines, is those cytokines are the second signal which is necessary to get activation of the B and T cells. So there are self-reactive B and T cells out in our periphery, but they are tolerant. They don't respond unless there's also this danger signal that results from a pathogen encounter with the phagocyte. Yes? Oh. Early developmental stages of the host. So early in fetal development of the mammal. So they're continuously being produced and they're continuously being selected. Now later, as we age, that process ramps down a little bit. But you're producing in your bone marrow self-reactive B and T cells now that are being knocked off or that are self-destructing now. Yeah, as you get older, what happens? So there are people that have their thymus taken out. Is that? It does. And so really, as you age, your repertoire becomes more fixed, and you're not producing these. You're not producing. As you age, that amount of new clones that are being positively and negatively selected are not generated as fast. Yes, we could talk about plasma cells, which are the antibody-producing B cells. So what are these B cells and T cells, and what do they do? Well, when they encounter the pathogen, right here, we've got very large numbers of shape recognition that's possible, but we have very small numbers of each. Too few to ever be able to affect clearance of the pathogen. And so when people talk about clonal selection, we're talking about the shape of the pathogen driving which T and B cells respond. And so as it overcomes this threshold of activation, whatever shape is seen by the very small numbers of B and T cells that are present, those B and T cells that respond to that shape are selected from that very large number of different clones, but very few numbers of each. And those numbers begin to ramp up. And when we go to look at what happens after those calm back down, is those specific clones that recognize that shape are now at a higher level and at an enhanced activation state following infection. And so one of the examples that is obvious about when we see these kinds of responses is chickenpox. When you first encounter chickenpox, all these elements of innate immunity and the ramping up of the B and T cells that respond to the varicella zoster virus that causes chickenpox takes a while to get going, and we actually get the disease chickenpox. But when we resolve that infection, and we have memory cells, memory B and T cells, that are specific for the varicella zoster virus, but are at a higher level and able to respond more quickly and efficiently to future infection, we now are immune to chickenpox. It's not that we're not getting reinfected with this virus. It's that when that pathogen is encountered again, the curve of that pathogen and resolution is so fast that we don't even recognize that we have the viral infection. Yes? And this is doesn't produce memory cells or loses memory. So there is what controls the loss of memory is an area of active study, but different pathogens appear to have to provoke different levels of initial memory responses, and the decay rate of those clones that respond to different pathogens appears to be different. And so for some diseases, we can see later in life a waning of immunity. Chickenpox is one that we do see. Later in life, people get shingles. It's the same virus. It's a different manifestation of that virus. But that's a waning of the natural immunity that people get through infection with chickenpox. There are some immunizations that we get that have to be boosted. So there's some immunizations that you have to get a booster more often than other immunizations. It's empirically determined. It's, how long do these seem to protect people, and we try to boost before that level wanes. And what boosting does is just do this again so that your memory response gets back up to where it could potentially be. Yes? Unknown. So probably a combination of both. It's pretty rare that people get chickenpox twice, and it is usually circumstances where the virus has gotten, for some reason, sequestered to one part of the body, and that the immunity, the memory level, is incomplete. So some of these immune responses take place in the lymph nodes around the area where the infection occurs. And if the virus hasn't spread to other areas, there is the possibility of reinfection, but it's really, really rare. As is, they now have a vaccine for chickenpox, and what that's really doing, it's a version of the varicella zoster virus that's been passaged in culture so that it doesn't cause chickenpox, but yet it looks to the immune system the shape of the chickenpox virus, and so it ramps up and comes down, leaving a memory response that is effective against the real chickenpox virus. And so there is, in some low levels of individuals, incomplete protection from that as well. It's just a different form of disease. It affects the nerve endings on the surface of the skin, and is a painful manifestation of this virus. It's a replication of the virus at the nerve endings. Rather than chickenpox, I am not completely certain. It has to do with the architecture of these viruses. Varicella zoster is a herpes virus. It hides in the nerve endings, and so it comes the first time. It presents differently than when it reemerges from the central nervous system back down to the peripheral nerves. Instead of an initial encounter with the virus from the outside, what shingles is a reactivation of the virus in the nervous system, and it passes down through the nerves to the nerve endings to cause shingles. So the source of the virus is different, and I think that is what is responsible for the difference in presentation. Some of that is unknown, but adults seem to get much more severe consequences of chickenpox than kids, and that may be a function of differences in the immune system as we age. So let's talk about what the B and T cells are recognizing. And so these are the effector cells of the adaptive immune system. And so they have different roles, and so why is it a B cell responds or why is it a T cell that is responding? And the differences have to do with where the pathogen is relative to the cells of our body. So there's some pathogens that get into our bloodstream and never get inside of our cells. And those pathogens that are outside of cells, and even those that eventually do get inside of our cells, have to at some point have been outside of our cells. They are free in the bloodstream, and it's the B cells and the molecules produced by the B cells that recognize those pathogens that are free in the bloodstream. And so what this B cell does is it has its immunoglobulin receptor, and it may bind a virus or a bacterium in the bloodstream, and in response to that, it expands in numbers, it proliferates. That's the clonal selection part. And then you have many different B cells with that same receptor present. And what the B cells predominantly do is secrete these receptors. They spit them out, and these are antibody molecules. They look like double-lined Ys. And what these antibody molecules can do is bind to the pathogen and block its ability to get into our host cells, so our own cells. So if you've got a virus or a bacterium that's trying to get inside of cells, it uses specific receptors on the bacterium and specific receptors on the host cells to get into the host cell. If you have antibody molecules that bind to these receptors, you block the ability of the pathogen to get inside of the cell. And so that's inhibition of entry. This binding up of the pathogen also can clump the organism. So these are bilobed Y molecules. They can form big chains of these because each of these parts are able to bind to the antigen or the pathogen. Antigen is the part of the pathogen that is recognized by the immune system. Yes? So they get chewed up in the bloodstream and filtered by the kidney. So they get chewed up through phagocytes. So phagocytes could take these up and chew them up. Things that are clumped also can get filtered in the kidney if the complexes aren't too big. And the other thing that these antibody molecules can do is bind a series of proteins. These proteins are known as complement, winding up with what's known as the membrane attack complex. The membrane attack complex can actually directly punch holes into the host, into the pathogen, destroying the pathogen. So these antibodies have multiple functions at being able to clear external to our cells, not external to our bloodstream, but external to our cells, bacteria, parasites, and viruses. So the B cells are recognizing the whole surface of the pathogen, or some whole protein on the surface of the pathogen. But this process is imperfect, and at some level, intracellular bacteria are able to get inside of host cells. They bind to a receptor, and then you wind up with a bacterium or a virus inside of the host cell. And once the organisms are inside the host cell, the antibody molecules can't do any good because they can't get inside of the cell. And so once a pathogen is inside of a host cell, it requires the T cells respond to get rid of the pathogen from inside of cells. And so if you have a pathogen, a virus, or a bacterium inside the host cell, it produces proteins or secretes proteins inside the host cell. These proteins are continuously being broken down inside the host cell. There's proteolytic enzymes in the host cell that break these proteins down into peptides, into small fragments of protein. And these are assembled in the cell onto special host molecules, molecules produced by the host, known as MHC molecules. And they're called MHC molecules because they're encoded in a genetic region of the host called the major histocompatibility complex. But we could just refer to them as MHC molecules. And so these peptides are continuously bound onto these MHC molecules and then displayed on the cell surface. And this process doesn't recognize that there's a pathogen inside of here. All proteins in all cells are continuously being broken down into peptides and displayed on the cell surface. So the vast majority of peptides that are found on the cell surface of all of our cells all the time are normal cellular self-proteins. And so why aren't the T cells responding to those? Because they've been deleted during education of the T cells in the thymus. So T cells aren't recognizing whole proteins. They're recognizing this peptide plus the MHC molecule. And if these MHC molecules have a self-peptide, we don't have the T cells in our repertoire to recognize these self-peptide, self-MHC complexes. That what a T cell recognizes as foreign is a non-self peptide in the context of a self-MHC class 1 molecule. And so again, if the T cell, if there's a virus in here and that virus is chewed up, the viral products are chewed up into peptides, those peptides are assembled on MHC molecules, and the T cell, through its T cell receptor, recognizes this foreign peptide in the context of the self-MHC molecule, the T cells will expand. They'll be clonally selected. And so what do T cells do? T cells have several effector functions, several functions that can deprive the pathogen of its intracellular environment within the host cell. One of those is direct lysis of the presenting cell. So in the case of viruses, they use the host cell as a factory to produce many different viruses. In the case of bacteria, they get in, they replicate inside of this host environment, avoiding the effects of the antibody molecules. And so if the T cell is able to pop this cell, it frees the virus before it can reassemble into new viral particles, depriving the organism of its replicative niche. The other thing that the T cells do, somebody mentioned the helper T cells. So these T cells that pop the host cells, those are known as cytotoxic T cells or killer T cells. There's also helper T cells. The helper T cells send out messenger molecules, cytokines, that call in more of the inflammatory response. More phagocytes into the area. And so the phagocytes aren't specific. They have no way of seeing self from non-self. They're just continuously gobbling up particles in the body and chewing them up. And so there's no specificity. It doesn't know whether it's a pathogen or just some piece of a normal cell that broke off. But once this signal comes off of a T cell, the cytokine signal comes off of the T cell, it brings in more of these inflammatory cells, the phagocytes, into that area of infection, lending to those less specific elements of the immune system some measure of antigen specificity. So that's the B and the T cells and how they expand and how they respond to infection in general. And then what we can do in the last few minutes is talk about how that works in terms of a specific kind of infection. Yes? Talk about the T cells first, but it's a good point. So the T cells, what we talked about was the first signal. The first signal is this T cell receptor recognizing the foreign peptide plus the self-MHC class I molecule. That's signal 1. The second signal are these co-stimulatory molecules that also must be present on this host cell. And so when we ask about, well, why aren't T cells that are free in the area recognizing toenail proteins that are chewed up in the peptides and displayed on the cell surface, that unless there's an infection present, unless this antigen presenting cell is also in the presence of cytokines that are induced by shapes that are typical of many different classes of pathogens, these co-stimulatory molecules aren't going to be present on the cell surface. And only if these co-stimulatory molecules are present do you get T cell activation. So it requires this signal and the second signal. B cells, just one second, then B cells are actually dependent on T cells in most cases for their activation. So the co-stimulatory danger requirement comes from the fact that the T cells also need to see this danger signal and that the B cells are dependent on these cytokines produced by the T cells as well as specific T cell B cell interactions in order for the B cells to completely ramp up. So there's an interplay between these two cell populations as well that keeps these cells from being autoreactive. So it can be stimulated by cytokines. It also there's other receptors on the surface of these antigen-presenting cells, the cells that chew up the antigens and present it to T cells. And these receptors recognize things that are usually present when one has an infection with a bacterium and a virus. It's particular, they're called pattern recognition molecules, and they recognize the fact that there is a general structure of a bacterium or the general structure of a virus present in the area.
So do any cells have that type of receptor? Because any cell could be harmful?
So what I have done here is kind of combined the two roles of antigen-presenting cells. So there are professional antigen-presenting cells, dendritic cells, macrophages, that do this co-stimulatory portion and ramp up the expansion of the B cells and T cells. That's the initial expansion. Once you have expansion of the B cells and T cells, those then no longer need the co-stimulatory molecules, and they can go on and recognize infection even in cells that don't have the co-stimulatory molecules.
And those professional antigen-presenting cells are part of
They're part of the innate immune system. They're the dendritic cells and the macrophages. Yes? Yes. Humoral responses are antibodies, things that are floating around in the bloodstream. The cell-mediated responses are these kind of responses, like the T cell responses that involve a cell-cell interaction as opposed to a free molecule, a protein pathogen interaction. So they're actually purifying, in the case of hepatitis, they're purifying antibodies from people who have been infected with hepatitis. Well, you get a specific gamma globulin shot for specific things.
So you are
And
doing specific.
that's called passive immunization because you're passively transferring somebody's already formed immune response, as opposed to giving an active immunization with a piece of the hepatitis virus that causes your own body to generate all of that. And so the passive immunization only really allows a small arm of the immune system to get up and going. Yes?
When you have organ rejection, something like organ transplant rejection, that's the B cells recognizing the antigens of the foreign cells?
No, it's the fact that the T cells really are recognizing a MHC molecule that's not self either. So, and that really has very little to do with how the immune system normally works. It just is a byproduct of how the immune system works when you try to take somebody's tissue and put it in somebody else. That wasn't, the immune system didn't evolve to cause, the immune system didn't evolve because that was a particular problem. It just is a byproduct of how it works. Yeah, it is possible to do that, but most of the time you're right, it's not done. And so what you're doing is taking serum from somebody that, because they've encountered hepatitis, has a higher, what's known as titer, a higher level of specific antibody against that organism. But you're also getting antibodies against other stuff as well. But they're just making sure that there's a certain amount of antibody that recognizes that pathogen. So in the last few minutes, I'll just give one example of how this works in terms of one bacterial pathogen. And so it's one of the organisms that immunologists have used to study how these processes work. It's a foodborne pathogen called Listeria monocytogenes. It is found in unpasteurized cheeses. It's been found in deli meats. It's found in seafood products that haven't been handled properly. And in most healthy individuals, it doesn't cause infection. It really causes infection in people that have underlying defects in the immune system. And that's why when we put it into normal animals, we can see how the immune system responds to this pathogen and clears the pathogen. The way it works when people eat contaminated food products is that the organism crosses through the epithelium of the gastrointestinal tract and gets to the cells in the liver, the macrophages that line the ducts that supply the liver. The organism is taken up there and it grows inside of the liver cells. And so what happens in the liver is that one cell, and the cells that line the ducts of the liver are macrophages. The organism is first free in the bloodstream, and at that point, antibody molecules can bind to the organism and block its entry. It works to some extent, but it's not completely protective against the organism getting into the macrophage, or the host cell. There are neutrophils in the area that are able to very effectively take up Listeria and do it in. And other phagocytic cells and other functions outside of the cell that can do in the Listeria. But at some frequency, it gets into the macrophage. It's taken up by the macrophage. And once it's inside of the vacuole, the phagocytic vacuole within the macrophage, these organisms have a protein that is activated inside of this vacuole. The pH drops actually inside of the vacuole. And when the pH drops, this enzyme produced by Listeria becomes active. It actually breaks down the vacuolar membrane, and these organisms escape the killing functions of the macrophage and become free in the cytoplasm of the host cell. These organisms are rod-shaped organisms. They're able to actually polymerize the cytoskeleton, the skeleton of the host cell, on one pole, causing these fibers to form that are made out of actin, one of the structures of the cell cytoskeleton. And it uses this polymerization of the actin fibers to actually have what it looks like a comet tail, and can project itself in a swimming motion all around the cytoplasm of the host cell. And at some frequency, it'll bash into the cell membrane and protrude that membrane. And this is an organ. In doing so, it invaginates the membrane of the neighboring cell. And at some frequency, this pinches off, and you've got an organism with the rocket tail behind it getting pinched off into this neighboring cell. As the endosome here acidifies, the organisms become free in the next cell. And so Listeria can spread cell to cell without ever being exposed to these things that are very effective at killing it. It actually enters a macrophage at first, and then it enters all the other cells of the liver, the hepatocytes, from there. And so it can spread throughout the liver. It's taken up by macrophages readily, and then it uses the cell-to-cell spread to get into the rest of the liver cells, the hepatocytes. And so this organism is able to grow and spread in the liver without being recognized by the antibody mechanisms. But what it's doing is, in its metabolism and to polymerize actin and to break out of the membrane, it secretes proteins. And these proteins are free in the cytoplasm of host cells, and they get chewed up into peptide fragments and assembled onto MHC molecules in the liver. And in someone with a normal immune system, the T cells are able to recognize these Listeria peptides in the context of the MHC molecules and pop those cells, depriving the organism of its ability to spread cell to cell without being recognized by the immune system. And the neutrophils do them in pretty quickly. And so if you infect an experimental animal, a mouse, with Listeria, you actually can't give it very many. So if you give it less than 10,000, the animal will recover from infection and live. You'll see that curve of the pathogen beginning to replicate and being cured off. But what's left here is a memory population of T cells that, when that animal recovers, you then can give it millions and millions of organisms, and it will recover from infection. So the neutrophils, it's not actually the T cells that are responsible for protecting this animal initially against death. It's the innate immune system, the phagocytes, the neutrophils. But the fact that the animal first encountered this pathogen and got rid of it allowed for the generation of a memory response that now can protect it from way more than 10,000 organisms. And so that animal is immune. And so one can take these animals that have been given this sublethal infection and take T cells from the spleens of these animals, one of the big lymph nodes. The spleen is, in our bodies, is here, and it's a lymphoid. It's a place where the T cells and the B cells migrate and encounter antigen. You could take that spleen from the animal, from the mouse, take out specifically the T cells, purify the T cells from that spleen, and put those T cells just inject them into a mouse that has never seen Listeria before. And if it hadn't received the T cells, it's going to be killed if the animal receives more than 10,000 organisms. But if it does receive the T cells, it's just as if it is just as if it had recovered. It's now immune to greater than 10,000 organisms. And so one can do experiments on these T cells to look at the kinds of proteins that they're responding to and the kinds of activities that they deploy to deprive the organism of its replicative niche. So there are other pathogens as well that do different things within the host cells. One of the other organisms that we study is Chlamydia trachomatis. So chlamydia enters into the genital tract or is introduced onto the conjunctival epithelium. It gets into the epithelial cells in those tissues. In the genital tract, you'd have cells lining the genital tract. These are individual cells on the surface of the mucosal surface. Chlamydia is able to bind to these and get inside of cells. Chlamydia, the infectious particle, can't replicate outside of cells. It only can replicate inside of cells. So once it gets inside of cells, it is again in a vacuole, and it replicates 100 to 1,000-fold within those vacuoles. Eventually, the vacuole is compromised, and the organisms are released to infect neighboring cells. Again, that initial infectious particle can be recognized by antibody molecules and neutralized for entry. But once these organisms are replicating inside of the cell, it requires that the T cells recognize peptides from these organisms in the context of the MHC molecule to break that cell open and deprive the organism of its replicative niche. So the goal of immunization is to look at what these T cells are responding to and what these antibodies are responding to, and develop methods to stimulate those T cells in the absence of real infection. And that's really what vaccines are designed to do. And the challenge is to figure out how to not only figure out what the T and B cells are responding to, but also to think about how you deliver those antigens safely into the bloodstream, which is actually less complicated than figuring out how you would deliver the antigens into host cells without damaging the host cells. And so the new set of vaccines uses things like viral carriers to deliver antigens from things like Listeria or Chlamydia or Salmonella or SARS into host cells to stimulate the T cells that respond to those so that you have an expanded population of T cells that could respond to them more effectively. A potentially very, very serious public health problem if we have a bad flu season and many of our people aren't vaccinated. The reality is that in years when the vaccine has been available, only a small percentage of our population really avails themselves of it anyway. And so I think there is some measure of when something is not available and it generates the publicity, it becomes more desirable. Exactly. And hopefully that desire will transfer to next season when there really is enough. The problem with it is that the problem with that vaccine that's different from most other vaccines is the fact that it's incredibly complicated to make because it's made in chicken eggs. It's not made by recombinant DNA techniques that most other bioproducts are made either chemically or made in giant fermenters in terms of recombinant DNA or recombinant cell culture products. And so the fact that we are still using technology from decades ago where you take these seed stocks of the vaccine and you inject them into eggs and let them grow in the egg and then purify the vaccine from the eggs just needs to be addressed. And it is in no way it's a complicated engineering problem, but it's no way something that can't be through targeted research towards developing better methods of doing that, where we could produce vaccine that is more responsive to the needs than having to come up with a huge number of doses and wondering whether anybody's going to use it. If we could produce it in a matter of weeks rather than in a matter of months, it wouldn't be as big a problem. Yep. What we have been talking about are acute infections, where what happens is there's this balance or this race between the pathogen and the immune system, and where the pathogen wins or the host wins. What a chronic infection is, things like tuberculosis, a bacterial pathogen, Mycobacterium tuberculosis, or HIV, a viral pathogen, these are chronic infections. And the immune system doesn't work the way that we see it working for acute infections during chronic infections. The presence of the pathogen long-term, what's going on in a chronic infection is a long-term balance between the immune system and the pathogen, that the replication of the pathogen is actively being checked by the ability of the immune system to suppress it. And for many, many years, that can be a tight balance that doesn't get disrupted such that the pathogen is cleared or that the host dies. And when the pathogen is around for so long, chronically stimulating the immune system, the immune system's, the ability to target the immune system with things like vaccines or boost the immune system becomes very complicated. In the case of HIV, it's infecting the T cells, dysregulating the function of the T cells. And so it has evolved very clever mechanisms of ensuring that the immune system isn't very effective at getting rid of it. And it is a big problem, and one that, unlike the flu vaccine problem where I say it's just an engineering problem, there's a fundamental intellectual problem with how one deals with a chronic infection where the immune system isn't responding normally. Thanks for coming.
My pleasure.
Thanks.